Good morning, everybody. Thank you for coming to my talk. Today I'll be talking to you about our work on spatiotemporal control of protein localization and intracellular metabolic flux with an RNA-based high-affinity dynamic scaffold. In modern metabolic engineering, we like to treat cells as mini factories. Most often, we use platform microorganisms to produce a wide range of chemicals. And the way we do that is we introduce novel metabolic pathways in these platform microorganisms. However, oftentimes, when we introduce novel metabolic pathways, they end up competing with the cell's native metabolism, which can yield low product titers or low cell growth. Traditionally, the way we get around that is through controlling the gene expression, so by overexpressing certain genes or deleting others, or by controlling the enzyme activity itself through site-directed mutagenesis and directed evolution of the protein. Oftentimes, however, these approaches require many iterations and do not give us the desired result. More recently, synthetic metabolons have been emerging as an alternative. Now, metabolons are temporary spatial organizations of enzymes that help increase metabolic efficiency by channeling substrates between enzymes. As you can see here, when the three enzymes are apart, the intermediates can diffuse out much easier. But when they are clustered together, you get a lot more of the final product through substrate channeling. There are multiple examples of metabolons in nature where the cell can assemble the metabolon to produce a desired metabolite when it needs to. However, there is one big difference between metabolons found in nature and synthetic metabolons developed thus far. And that is that synthetic metabolons are only static, which means that they can only assemble once, whereas in nature, the cell can choose when to assemble and disassemble a metabolon based on its metabolic needs. And why is that a problem, you might ask? So in native metabolons, there are feedback loops based on the sensing of substrate product or cofactor concentration that signal the cell when to assemble and disassemble them, and that helps prevent metabolite imbalances inside the cell. However, with the current synthetic metabolons we can still only redirect metabolic fluxes once, which may work in a lot of cases, but in other cases you might end up with undesirable metabolite imbalances, and that's what we want to avoid. That's why we want to develop a dynamic scaffold to control this metabolic flux. So now that we've established that we want a dynamic scaffold, how do we actually achieve that? For our dynamic synthetic metabolon, we propose to use RNA strand hybridization to drive metabolon assembly and toll-mediated strand displacement technology to drive metabolon disassembly. Just a quick refresher on what toll-mediated strand displacement is. It's a reaction between three different strands of nucleic acid. So if you start with two nucleic acid strands initially hybridized in the 1,1 star region, and then introduce this trigger strand to the mix, what you end up with is the trigger strand will initially hybridize in the 2,2 star region, also known as a toehold region. Following that initial hybridization, it will begin to displace the top strand and eventually completely displace it, thus resulting in the trigger strand hybridized fully with the bottom strand and the top strand completely dissociated from its original hybridization region. The reason this technology interests us is because it has a very fast time scale in the order of minutes to hours, and even more importantly, you can tune the kinetics finely to however fast you want it to happen. So by changing the length of the toehold and adding mismatch base pairs inside the toehold, you can end up with a wide range of kinetics. So you can make small changes in your system to very finely control how fast it'll associate and disassociate. So now that we've established how we want to make our metabolon dynamic, the next question is how do we ensure that these RNA interactions are the only driving force between the assembly and disassembly of the metabolon? And for that, we need a way to localize our proteins on these RNA strands with very high specificity and affinity. And for that, we've chosen to use CRISPR-Cas type 1 Cas6 proteins. Now, Cas6 proteins are small endoribonucleases of about 24 kilodalton in size. They are part of the broader type 1 cascade system, and they bind to short hairpin RNA motifs of about 10 to 20 nucleotides in size. Once they bind to this motifs, they eventually cleave the 3' end of them. They are of interest to us because they are proven fusion partners, have very low dissociation constants with the range going as low as 50 picomolar, and most importantly, there exist orthogonal Cas6 proteins, each with its own distinct binding motif, and they have very limited crosstalk between one another. So you can imagine, for example, if we were to express a long RNA sequence and then have 3 different distinct binding motifs corresponding to 3 Cas6 proteins, then each one would localize to its unique binding motif, cleave at the 3' end, and generate this small RNA molecule with a protein tightly bound to it. And that is what the building block of our scaffold is. So the way we propose the scaffold will assemble is you have your 2 orthogonal Cas6 proteins fused to 2 enzymes of interest, and then you express a long RNA scaffold molecule with two distinct RNA binding motifs. So the first thing that will happen is the Cas6 proteins will bind to their respective RNA hairpins. Following that, they will cleave the 3' end and generate the pieces we were talking about in the previous slide. Now, if you design these pieces to be complementary to one another, then you can basically just have RNA strand hybridization that will bring together your 2 proteins of interest and thus assemble the scaffold. The next step is scaffold disassembly. And to do that, we simply need to express a trigger, which through toehold-mediated strand displacement will begin by binding to the toehold and then eventually dissociate the top strand from the bottom strand, thus pulling the scaffold apart. So now that we've established how the scaffold should work, the next question is how do we quantify this level of assembly? And for that, we want to use a split luciferase reporter system. Split luciferase reporter system is comprised of the small bit over here and the large bit over here. The 2 apart have very minimal activity. However, when they are brought into close proximity with something such as our scaffold, we will see an increase in luminescence. So we can measure that luminescence and use that as evidence that our scaffold is actually assembling as expected. Now we put all these components inside bacterial cells and we decided to run these assays over 8 hours and measure the luminescence. So this is the fold increase in luminescence as measured by this ratio over here, where we have samples where we express both the proteins and the scaffold RNA, or when we only express the proteins. And you can see that at 2.5 hours, there is about a 10-fold increase in luminescence between these 2 samples, which is pretty promising. But even more impressively, when we go to 5 and 8 hours, you see an even more significant uptick in the fold increase in luminescence, peaking at about 220-fold at 8 hours. That tells us that if we express all the correct components of our scaffold, it will assemble as expected. And furthermore, it is a stable enough scaffold that it actually begins accumulating in the cell, hence the large increase in luminescence over time. Having seen these promising results, the next question is, can we disassemble the system using an independent inducible trigger? So as you can see in the cartoon here, we basically want a scaffold that will be able to dissociate in the presence of the correct trigger sequence. To examine this, we added an independent inducible trigger in our system. So we expressed our regular scaffold RNA, and then we expressed one of 3 trigger constructs. So the first one is a fully scrambled trigger that should not disassemble the system. The second one is a scrambled toehold trigger, which should not disassemble the system if the main driving force behind this disassembly is toehold-mediated strand displacement. And finally, we have our regular trigger, which should disassemble the system if everything is working as expected. So we ran our experiments, and over here we have the luminescence ratio, which was calculated by taking the luminescence of proteins with scaffold and trigger expression over proteins plus scaffold only. So you can see that at t equals 0, which is the time we induce the trigger, all of our samples have a ratio of about 1, which makes sense since at that point there is no trigger to cause any dissociation. But then 5 hours post-trigger induction, we see both for the fully scrambled and the scrambled toehold trigger that the ratio actually creeps up, indicating that there's no significant change when we express these scrambled trigger variants. So our scaffold is not actually dissociating at all. However, if we look at the regular trigger instead, we see that 5 hours post-induction, we actually see about a 50% drop in luminescence in the sample where we expressed the trigger versus the sample where we did not, indicating that our system is in fact disassembling through toehold-mediated strand displacement thanks to the expression of this independent trigger. So far we have shown that we can assemble a scaffold and then disassemble it with the expression of a trigger. The next question we wanted to ask was, is there any way for us to turn on the assembly using a trigger? So instead of disassembling, we assemble by the induction of an independent trigger. To do that, we had to modify the architecture of our system so that the trigger actually induces scaffold assembly. And to do that, what we did is, for our first RNA piece over here, instead of having the 5' overhang exposed, we made it kind of hybridize onto itself with a hairpin strand, which can then be unblocked by the expression of the correct trigger sequence. Once it is unblocked, this exposes the 2' region onto which the 2 region of the second part of our scaffold can now hybridize to. And this leads to the scaffold assembly as we expected it to. In order to test this, we had a very similar system, except in this case, we constitutively expressed the 2 pieces of the RNA scaffold separately. And then to that, we added the inducible trigger that was either, again, a fully scrambled trigger, which should not assemble, scrambled toe hole trigger, which should not assemble if the main driving force is toe hole mediated strand displacement, and finally a regular trigger, which should assemble if everything is working as expected. So we ran the similar experiments, and again we have a fold increase in luminescence here, which is taken by this ratio as shown over here. And we can see that the regular trigger behaves very similar. When we express the regular trigger, the system behaves very similarly to when we had the original system we had designed, peaking again at around 220-fold increase in luminescence. So that is working exactly as we expected to, and as well as the original system we had. On the other hand, the scrambled toe hole trigger and the fully scrambled trigger show some increase in luminescence, but much less than the regular trigger, indicating that we might have some non-specific activation because of the nature of our system, but it is nowhere near that with the regular trigger. So overall, we would say that the turn-off system assembles at similar levels to the turn-off system only when the correct trigger RNA is expressed and through toe hole mediated strand displacement. So having both our turn-off system and our turn-on system, the next kind of thing we wanted to question is, can we actually use these systems to increase product titer in a metabolic pathway? To do that, we wanted to select an appropriate pathway for our first test. So we wanted a 2-step pathway where the proteins are monomeric and the product can be quantified relatively easily. And for that, we chose this indole-3 acetic acid, which is a platform chemical with many downstream uses. Indole-3 acetic acid is produced in vivo through this 2-step pathway where tryptophan goes through IAAM to indole acetamide, and then through IAH to indole-3 acetic acid. So we fused our IAAM protein and IAH protein to the 2 orthogonal Cas6 proteins and ran 16-hour fermentations with both the turn-off and the turn-on system, and then analyzed the supernatant on an HPLC. We had 2 different protein expression regimes, where we had low protein induction and high protein induction, just to see what the different protein levels would behave like in our system. So the first thing we would like to note is that, in fact, both the turn-on and the turn-off system behave very similarly, showing similar increases in final titer of our product. For the low protein regime, we saw that expressing the scaffold results in about a 9-fold increase in indole-3 acetic acid titer. Whereas for the high protein one, we see about a 3-fold increase in product titer. So in both cases, we see a very significant increase in product titer thanks to our scaffolding. With the lower protein, it's a bigger one. It's a bigger increase because of the lower background. Finally, the most important kind of point we would like to make is that we see that the low scaffold protein plus scaffold yielded very similar titer products to high protein without any scaffold, indicating that you can actually get the same amount of product as if you had a lot more protein just by having the scaffold. So we can minimize the metabolic burden of expressing these non-native proteins by just simply using our scaffold and get similar productivity from our cells. So our scaffold system can be successfully increase product titer. In the future, we would like to use native RNAs and mRNA as trigger strands. We would also like to implement feedback loops based on metabolite sensing to tell the scaffold when to assemble or disassemble. We'd also like to develop a recycle system that allows multiple transitions from assembled to disassembled states. And finally, we want to expand scaffold capabilities to more complex pathways. Just to conclude, I talked to you about a dynamic high-affinity modular scaffold toolkit, which uses RNA hybridization and toe hole-mediated strand displacement to drive scaffold assembly and disassembly. And we use Cas6 proteins to localize these enzymes of interest with high specificity. And we want to do this to prevent metabolite imbalances and introduce a new toolkit for optimizing non-native pathways. With that said, I'd like to thank Dr. Wilfred Chen, the Chen Lab, and our funding sources, and I will happily take any questions.